All right, welcome to another Build a Cell seminar. Thank you everyone for being here. And I'm going to toss the ball over to Yuval to introduce himself and tell us more about what you've been working on. Thank you, Yuval, for agreeing to give a seminar speech.
Yeah, sure thing. So thanks very much for having me. Thanks for the invite, Kate. Yeah, thanks for everything you've been doing for the Build a Cell community. It's fantastic to see this sort of community initiative every few weeks, people gathering, exchanging ideas. Yeah, it's amazing to see. So my name is Yuval. I'm a Future Leaders Fellow and an Associate Professor at Imperial College London in the Department of Chemical Engineering. And yeah, I'd like to talk to you a bit about what our group does in this field of synthetic cells and related fields. So I guess by virtue of you guys being in this call, you probably know what a synthetic cell is. You know the key themes around synthetic cell research and the motivation. So I'm not going to dwell too long into this, but briefly speaking, high-level overview, synthetic cells are structures constructed from the bottom up that mimic some feature of a living biological cell. And they're very useful as models to understand biology in a minimal environment, but also very useful as engineered devices for potential use in biomedicine, in sensing, in environmental remediation, and so on. Generally, they're composed of some sort of capsule, some sort of chassis. In our group, we're very big on lipids and membranes. So we're very good and very interested at engineering membranes. And our idea, one theme that we come back to again and again is that a membrane is, most people think of a lipid membrane in the context of vesicle of synthetic cells as just a chassis, just a capsule which defines the inside and the outside of a cell. But we actually think that membranes themselves can be used as very interesting functional components in a synthetic cell. So if you want to design a synthetic cell that can swim up a concentration gradient and can sense and respond to its environment and so on, yes, you can use DNA and proteins to give you that sort of sophisticated behavior. But we think that by engineering functional membranes, the membranes themselves can also be used to impart functionality such as motility, sense and response, and so on. And that's something I'll touch on throughout this talk. Okay. I'd like to give one maybe slightly different take on why we think engineering synthetic cells is a useful thing to do. So very often, when I speak to people, I meet people in bioengineering departments and life sciences departments, and they, for decades, have been engineering bacteria or engineering yeast or mammalian cells and getting them to do things that they wouldn't normally do. It's quite a mature research area, genetic engineering, metabolic engineering, and so on. And one thing they want to know is, well, hold on, why do you want to go bottom up? Why do you want to start from scratch? Why bother going through this whole thing from bottom to top when you can just re-engineer a bacteria and get it to do something useful? After all, we all know that living cells are super, super complex and powerful as it is, okay? So there are many answers to that question, but one answer that came to me a year or so ago when I was sitting on the in the London Underground. I don't know if you can see this image here of a tube in the background. I was reading this book by Peter Thiel, and Peter Thiel, for those of you who don't know, is basically like he's an Elon Musk style character. He's like a multi-billionaire. He founded PayPal and a whole host of other tech companies. Um, and he's a very sort of controversial sort of figure, very opinionated figure, but he's one of the most successful business guys and serial entrepreneurs on the planet. And he basically gives a series of lectures to Harvard Business School, and he compiled those lectures into a book. And so it's a really good book. It's a really interesting read, and it's called Zero to One. And it's basically a book all about how to start a multi-billion dollar company. What do you need to go from 0 to 1 to change the world and to make $1 billion company? In any case, this book is not very, maybe 100 pages, but I'm not joking. Half of that book is dedicated to him saying, whatever you do as a potential entrepreneur, do not start a biotech company and do not invest in the biotech company. It's the worst thing you could do. He really hates biologists. He really hates life science startups. And the reason he gives for that piece of advice, which he devotes half that book, is as follows. So we all know what Moore's law is. Okay, Moore's law is basically what powers the modern economy and what has been powering the modern economy for the past 30 years, is this principle of this our exponentially increasing ability to compute, to process information on a microchip, and basically is the reason why computers are getting better and better and better and smaller and smaller and smaller. Okay, the number of transistors, technically speaking, Moore's law is the number of transistors in a microchip doubles every 2 years, and it's an exponential feature. Okay, but he basically says in biology and in biotechnology, I should say, it's not Moore's law, it's Moore's law but read in reverse. So it's Eroom's law, and it's basically inverted. In the biotech, he gives an example of the number of drugs approved per $1 billion of R&D spend has halved every 9 years. So it's getting harder and harder and harder and more expensive every year to work to bring a drug to market, and more generally to work and to succeed in biotechnology in the biotech space. And he says basically this trend's been going on since the 50s, and it's not getting any better. If anything, it's getting worse. Okay. And the reason for that, he basically says, is because you're working with living systems. Okay. And then he has a nice little table where he compares and contrasts a biotech startup to a software startup, which is the space that he works in. So in biology, you're working with living systems that are highly uncontrollable. For those of you who have tried to predictably reprogram or re-engineer bacteria to do something new, it's basically impossible. The bacterium will always sort of fight up against the genetic engineering which you impose in it. There's genetic drift, and they're basically uncontrollable. If you compare it to a computer, it's basically uncontrollable. Okay, with software, everything's known, everything's perfectly understood, everything's determinate. You're working with code. Same with the environment. A bacteria or a living cell is poorly understood. Impossible. I personally think it's impossible to fully understand what every molecule in a bacteria is doing or a living cell is doing. It's far too complex. You have probably somewhere in the order of hundreds of thousands of different molecules. Every molecule is interacting with one another, forming this humongously complex network space of interaction. So you'll never ever be able to understand exactly what every molecule is doing. And also from an engineering point of view, if you want to re-engineer one part of that space, you have no idea how the other part of that space will respond. It's very, very difficult, if not impossible, for us to properly engineer a living cell. Software, everything's well understood, everything is artificial. Regulation, okay? Heavily regulated. Biology is heavily regulated. If you want to re-engineer bacteria to sense a toxin in a lake, and you want to put that engineered bacterium in a lake, the chance of that getting approved is infinitesimally small. There are all sorts of regulatory hurdles. The bacteria could mutate. It could all have all sorts of unintended sort of consequences, which is why genetic engineering as a concept is heavily regulated. With software, it's basically unregulated. And that then translates to cost. Biotech, extremely expensive, software, extremely cheap. Only very little seed money can lead to a huge company. Okay. At the bottom, he takes it a bit too personal for my liking. Yeah, biotech startups, highly salaried, unaligned lab drones, and software's committed entrepreneurial hackers. So, when I showed this to my team, they got very offended, and rightly so. That was a cheap dig. In any case, synthetic cells, if you think about it, is basically a way for us to bridge this divide. Synthetic cells are not uncontrolled organisms. They're fully controllable. You know exactly what they're composed of because you make them from scratch. You can control each and every component of them. Everything's well understood. Everything's artificial. It's more close to engineered code to unengineered and uncontrollable organisms. Everything's definite. You can be a classical engineer if you want because you know everything to precision on a molecular sort of level. Regulation is, as I said before, it's basically unregulated in the sense that synthetic cells do not grow, they do not divide, they do not mutate. So that massively sort of lowers the sort of regulatory bars that you have to go through to bring a potential synthetic cell product to market. And we expect these things to have an impact in terms of cost and so on. Okay, so I think this maybe is a new perspective of why synthetic cells can be useful in a biotechnology context. It allows you to do things that you simply can't do with living cells. Okay, so onto the scientific bit of my talk. As I said, we're very big on membranes, and we're very interested in building different types of membrane architectures to mimic some of the membrane architectures that you see in cells. We are very interested in translating that architectural complexity into behavioral complexity. So taking these architectures and leveraging those to engineer new types of behaviors. That's the second part of the talk. And the third part is really starting to play around with different length scales. So for those of you who I guess, work with normal with synthetic cells, you're used to working with synthetic cells on the micron length scale, 5, 10, 20 microns across. But we are starting to play around with building synthetic organelles on the nanometer length scale because they're very useful for therapeutics and so on. And that's the third part of the talk. Okay, so in terms of architectures, so on the screen here, you'll see a whole host of different architectures that over the past 5 or 6 years, we've managed to build in the lab. Okay, so most people, when they think of synthetic cells, they think about this architecture on the top left, if you can see my pointer, a lipid vesicle, okay, a single membrane structure. But we can now build multi-membrane structures, for example, of a membrane within a membrane within a membrane. We can encapsulate large compartments inside the vesicle. We can encapsulate many small compartments inside a vesicle. It's called a nested system. We can link up individual synthetic cells through double membranes, something like gap junctions. We can link up individual cells through nanotubes, and so on and so forth. So we could do that. And importantly, we can do that with control of all relevant features. So what does that mean? So we can control the size. We can produce very monodispersed vesicles of a very defined size, all the way from the micron regime to a nano regime. We can control the lipid composition. So if you look here, a blue membrane, a red membrane, a black membrane, these are membranes of different lipid composition. We can control the lipid composition of each membrane in the system, as well as the cargo in each compartment. So like, like a blue cargo and a green cargo, different molecules, different components, and so on. So we can now do that. Now, what allows us to do that mostly is microfluidics. So we have a host of microfluidic technologies to allow us to form these different architectures. And I'll talk you through a few of those in the next slide. So microfluidic technology is really an underpinning technology throughout our lab. And in most cases, we work with droplet microfluidics, where each droplet is a template around which these membranes assemble. Okay. So one of the systems that we have is this microfluidic device to form to form giant vesicles. So this is like the base technology, and which feeds into many of the other sort of technologies. So there's the Euler method. It wasn't developed by us. It was developed by Kees Dekker's lab and Deschwande's lab when he was in Kees Dekker's lab, and other people, including Ulrich Kaiser's group, have also further developed this method. And it's basically you form a double emulsion. So it's a water and oil and water droplet. And by playing around with the physical chemistry of this thing, this middle oil droplet will de-wet. It will bud off from the system, leaving behind a lipid vesicle. And this is what it looks like on the microscope. You can see we're forming many of these vesicles in high throughput. We can form hundreds of thousands of vesicles a minute, and you can leave this thing running for hours and hours on end. The important thing here is a GIF of, we're zooming out to a population. You can see we're forming many vesicles. They're all of the same size, all very monodispersed, very high encapsulation efficiency, and the same fluorescence, so the same encapsulation efficiency, so very uniform population. Now, I'm not going to go into microfluidics because it's not the focus of the talk, but microfluidics allows us to trap vesicles, to analyze them over different timescales, hours, minutes, seconds, and so on. You can purify them from the surrounding solution. You could, and we are developing devices to make libraries of different synthetic cells, of different lipid compositions and different encapsulated compositions to do things like directed evolution and so on. But we very consciously design all our microfluidics to be modular. Okay, so we have a generation module, and we have a mixing module, and we have a trapping module and a perfusion module, and you can basically drag and drop and mix and match these devices for your experiment. So if any of you want to learn some microfluidics and take some of our modules and plug them into your devices, we're very happy to collaborate on that front. Another type of device here is another sort of device early on, which we use to make what are called multisomes. It's basically an assembly line, and it's an assembly line where in the first part of the assembly line here, you form a white droplet and a black droplet alternating, and then they flow through this meander, and then they get encapsulated in the second junction, and this is the product. Okay, so this is a multi-compartment synthetic cell, and you have lipids in the solution, so you basically have a lipid between the internal droplet and a lipid between the internal droplet and the external droplet. And this is actually Hagen Bailey actually was the first to demonstrate that these things can be formed by pipetting, but we've developed the microfluidics method to make them in high throughput. Now, the thing about these is that because there's a membrane there, you can put transmembrane protein in. You can shuttle things between the compartments. You can have information exchange between the inner compartments and the external compartments, and so on and so forth. So this is an example of a different type of microfluidic geometry, and by playing around with different device designs, you can form different architectures, as I've shown here. Okay, so we have our synthetic cells. One of the things we're also very big on is stimuli response. Wouldn't it be good and cool if you could design a synthetic cell that you can control from the outside, turn on and off and on and off externally, or a synthetic cell that can sense this environment, that can sense the presence of a disease state, turn itself on, produce a therapeutic, release a therapeutic, sense that disease state isn't there anymore, and then shut back down and become dormant again. Okay, these are the sort of things that we're thinking of. So one of the things we wanted to design is a synthetic cell that can respond to stimuli through the production of a protein. And the first stimuli that we wanted to show was temperature. And this is a collaboration with Francesca Cironi, with a PhD student at the time called Carolina, who just graduated. So what we did was we encapsulated a cell-free expression system in our synthetic cell. As many of you are probably aware, you can produce proteins from DNA template. But the thing that you produce is an RNA thermometer. These are RNA constructs which are engineered, and they're basically at a room temperature, they sort of zip up. And they basically block the access of the ribosome to the ribosome binding site. And at a very specific temperature, and at a temperature that's defined by the base sequence of this thermometer, these things can unzip, and you can get of a protein. So that allows us to call the translation of a protein with temperature. It was actually taken, this design from Petra Schroeder's lab, and we incorporated this into a vesicle. So you can see different temperatures, we can activate the cell-free expression at different temperatures by playing around with the bases. So you can produce green fluorescent protein in our vesicles, and you can see at different temperatures, you either do get expression or don't get expression. And we typically do, it works in a bulk well plate format. It works when you do analyze individual vesicles on a microscope, and that's shown in these graphs. So we've shown that you can produce a protein inside it with under the control of different temperatures using RNA thermometers, but you can also release. So you want to produce a drug, let's say, and then release it also. So to do that, we've shown that we can produce a protein, alpha-hemolysin, or as it's produced, alpha-hemolysin props into the membrane and it releases whatever's inside. Okay, and we've shown that. So when you add alpha-hemolysin to the solution, you see this red, in a flow cytometry, this red curve here starts shifting, and you start to get this emergence of this yellow curve, which are basically empty vesicles, showing that this assay sort of worked. And on a microscope, you start seeing these fluorescent vesicles, one by one, start to release their cargo. As alpha-hemolysin's done its thing. So these vesicles are still there. So they're there on the bright field, but they're just depleted of their fluorescent cargo. Okay, anyway, so that's an example of how we use cell-free expression systems, and we started to build more interesting and complex stimuli responsive synthetic cells. Okay, so how do we translate some of this architectural complexity that I've shown in the beginning to functional complexity? Okay, so I'm not going to, so just to give you a brief series of highlights from the group. One example is motion, hapotactic synthetic cells that can crawl on concentration gradient from a low concentration of ligand to a high concentration of ligand. And this is done in collaboration with Lorenzo Di Michele's group, and it's on the archive. So that's movement across a concentration gradient through sort of crawling-like motion. The second is signaling cascade, synthetic cells that can transmit signals from one compartment to the next through proteins, just like a living cell does. We've built synthetic cells that can do that. Communication. We've designed synthetic cells and synthetic organelles and can communicate with bacteria, back and forth communication, using stimuli responsive systems, either light or temperature. You can turn on this communication on and off. Protein synthesis, production of different proteins in different compartments, is something that we can routinely do. we can build exosomes on the surfaces of synthetic cells, release them in some sort of stimuli, and then these exosomes can then go on and infect and interact with neighboring biological cells. We can build gap junctions, synthetic cells that can link up to one another to form the basis of tissues. Again, through all sorts of fancy membrane engineering, we can take some vesicles and link them up to one another, and they form these gap junctions. You can see they zip together, and you can get communication and material transfer between compartments and so on and so forth. And you can also encapsulate living cells into our synthetic cells. You can encapsulate bacteria, you can encapsulate chloroplasts, you can encapsulate all sorts of living organelles into our synthetic cells to form hybrid systems. And I see Emiliano and Paola might be on the call. It's an active project with them to do these sort of fun things. Very recently, we showed that you can form the basis of a synthetic nucleus. So we can encapsulate a vesicle within a vesicle, a nucleus-sized compartment inside a cell-sized cell. And we can control the composition of each compartment. So you can actually start to load, for example, the DNA in the inner compartment and have other things in the cytosolic compartment and start to step by step build up complexity, build more and more architecturally complex eukaryotic cell models. And we've developed technologies to do that. We can, for example, have communication. Again, the constant theme in our group is communication between compartments. So you could have transcription in one compartment. You can produce proteins that pop into the membrane, and you have communication between the nucleus and the cytoplasm, and have activation of a protein in the cytosol compartment. So we want to start to approximate what we see in eukaryotic cells. Now, this is an example of what an experiment looks like in that front. Okay, so I'll talk to you about hapotaxis, which is this process of a synthetic cell crawling on a surface. But can we do chemotaxis, synthetic cells that can swim, for want of a better word, upper concentration gradients? And again, this is a nice example of how architecture and compartmentalization is used to achieve that. So in this case, the cells are not lipid vesicles. They are aqueous two-phase separator systems. They're basically peg-dextran systems. So the blue droplets was shown here in a blue droplet in a green background. But on the interface, on the surface of that two-phase system, you have liposomes. So you do have membranes on the surface. It's what's called a Pickering emulsion. They act like amphiphiles and they stabilize the surface. And we designed a system whereby if you apply polymer gradients, only one half of that synthetic cell gets destabilized. And when I say destabilize, I mean those liposomes on the surface detach. And as they detach asymmetrically, you then have an asymmetric surface tension gradient on the droplet, which then leads to motion due to the Marangoni flow. And this is what's shown here. So here's a whole population of synthetic cells. They're fluorescent because they have fluorescent liposomes on their membrane. We apply a gradient going from top to bottom, and you see very quickly you get a motion of the cells from the bottom to the top over large distances. So this is a millimeter basically. And you can see what will happen, the transition. So have a look at what's happening to the membrane. They start off uniform in terms of the fluorescence, but then they become Janus. They become asymmetric as these membranes peel off. And then this is what leads to this motion. So look at this one, the big one. You start to get peeling off of the liposomes from the top to the bottom. And at a certain threshold, they just shoot up, leaving behind a trail, almost like a comet, a trail of fluorescence liposomes. This is an example of how we can engineer some form of chemotaxis in our synthetic cells using principles of membranes and compartmentalization. Okay. Back to this theme of stimuli response. So I showed you how you can use genetics, i.e. RNA thermometers, to design a stimuli response to a synthetic cell. But there are certain things that are very difficult to do with genetics. Okay, so what sort of stimuli responsive cells are we and others interested in? So we want to design synthetic cells that can respond to different pH, hypoxia maybe, biomarkers. These are all things that are found in disease states. You can think of maybe designing genetic systems that can respond to these things. Intrinsic cues. So these are things that are, let's say, present inside the body in the target site and by the target site. You also might want to design a synthetic cell that's responsive to external cues, things coming from the outside, temperature, magnetic fields, and ultrasounds. So some of these things are very, very difficult to engineer using genetics alone. Just take magnetic fields. Okay, if you want to engineer a bacteria, which can respond to a magnetic field by producing a protein, is very, very difficult. Biology doesn't do magnetic systems very well, very easily. It's probably doable, but it's a very, very challenging thing to do. But with synthetic cells, they're completely synthetic, completely artificial, and you could very easily plug in non-biological components and building blocks. So you can very easily put in a magnetic nanoparticles into a synthetic cell, and then it works. And that's what we've done to design a magnetic responsive synthetic cell. So this is an example of a typical system that we use. So we have a nested vesicle. So it's a synthetic cell with many smaller organelles within it. And this external membrane is completely inert. It's not responsive to anything, this red membrane. And inside, you have these blue organelles, these blue membranes. And these blue membranes are stimuli responsive. So they normally will house a reagent. They can house, for example, a substrate or an inducer molecule. And outside these organelles, these blue dots are the enzymes in this case. So this whole thing's dormant because the enzyme doesn't see the substrate. But as soon as you hit it with a stimuli, it could be temperature, it could be light, it could be magnetic fields, it could be anything. These pores open up inside the organelles and they release the inducer molecule or they release the substrate, and you can activate the synthetic cell externally. And in this case here, as a proof of concept, you see a fluorescent molecule being acted on by an enzyme to produce a fluorescent molecule, okay? So basically, the question then becomes, how do you design magnetoresponsive membranes, membranes that release their cargo in response to the magnetic field? So what we do is we take membranes and we take membranes that are thermoresponsive. So when you take a membrane through a gel to fluid transition, at that transition, for various interesting biophysical reasons, they become permeable. And if you then attach magnetic nanoparticles on the surface of these nanomembranes, and you apply an alternating magnetic field, they very locally heat up the membranes, leading to content release, leading to activation of a enzymatic reaction, leading to fluorescence. Okay, and that's basically how we design our magnetic responsive cells. So we've built and we've used these heating magnetic induction systems. You pop your samples in, you turn it on, you take the membrane, and only the membrane, because it's localized to the membrane, through a gel to fluid transition, and you activate the system. And this is what it looks like. On the microscope, only with the magnet on and with the nanoparticle present do you get fluorescence, you get activation. This is what it looks like on a population level, with and without the magnet. And you can basically tune it at different magnetic field strengths. You can get more and more activation of our synthetic cell. So that's an example of how we can use magnetic fields to activate synthetic cells. We apply the same sort of tricks. You can get light activated synthetic cells by working with photopolymerizable lipids. So these are lipids which basically hit it with light. These triple bonds here polymerize. They scrunch up to form a grain boundary. And that's how you have light responsive organelles and temperature responsive organelles. Similar to what I said before, you take a membrane through a phase transition, which you can see on a microscope sometimes, and you can activate a process within. Okay, so the final part of my talk is, how do you move towards the nanoscale? So most of our synthetic cells are size of a bacterium or size of a mammalian cell, maybe 5 microns or bigger. But for many applications, you want to go nano. Okay, so think about therapeutics. In traditional drug delivery, they say you need to have lipid nanoparticles and liposomes, which are maybe 200 nanometers, for them to penetrate deep inside tumors, to navigate through the capillaries, to get through the blood-brain barrier, and so on. So it's a big challenge. So one is a synthetic cell, which can encapsulate all those materials in such a small compartment, but also just a classical liposome, a lipid nanoparticle, okay? So we asked our question, right? We've been very, very good over the years to form compartments in membrane compartments on the micron scale. Can we form membrane compartments on the nanoscale to do something interesting? Again, we know from biology that compartmentalizing content and membranes and compartments give cells functionality. It's the sort of stuff that you read in GCSE or, in high school textbooks. Why is a bacteria less sophisticated than a eukaryotic cell? Because eukaryotic cells have compartments which exist in different chemical states and allow them to do different things, and so on and so forth. So if you look at a lipid, a very structurally simplistic. A lipid nanoparticle is just a solid blob, and a liposome is also just a blob. It's just like a spherical compartment with things inside. No architectural complexity at all. Can we start to build architectural complexity into these lipid-based particles, and can these be used in synthetic biology? So to do this, we turn to microfluidics again, but in this case, instead of droplet microfluidics, we use a technology called hydrodynamic flow focusing. So everything's continuous. So there's no droplets there because droplets, there's a limit to how small you can form a droplet. Okay, you can't form a nano-sized droplet using traditional droplet microfluidics. So we formed a system which is a bit more reliant on self-assembly. It's pretty simple, right? So if in one stream you have lipid dissolved in ethanol, lipids are soluble in ethanol, and in the other stream you have an aqueous stream, and you flow them through, you basically have diffusive mixing. So over time, as you progress through this channel from left to right, you get mixing of the ethanol in the aqueous buffer. And at a certain point, you'd reach a critical concentration of ethanol, at which point the lipids just stop being soluble in solution because you're diluting it with water, and they're not soluble in water. So they start crashing out, I guess, out of solution, and you get self-assembly of particles, but very, very well controlled. Okay, so by controlling the flow rates, you can control the features of this interface, and you can control the size very precisely of these particles. So we can form different types of particles. These are very intricate, very beautiful, architecturally complex particles. They're basically mathematical minimal surfaces of various different symmetries, various different space groups with a lipid bilayer sort of draped around them. In this case here, they're called cubosomes, of cubic phases or hexagonal phases, different types of phases you can form interconnected lipid networks of very defined order. And by changing the lipid chemical structure, you can form these different particles, essentially. And this is what these particles look like on the microscope. Very, very well ordered. You can analyze them, the cryo microscope, or using a synchrotron source to determine what phase you have. And you can load DNA in these phases. You can load small molecules, proteins in these phases. But as I said before, the important thing with microfluidics is it allows you to very precisely control the size. And that's what you see on the right here. You see going from changing the flow rate ratio, so the ratio between the different inlets, the streams, you can control the diameter. So you can see here, this blue, you can go from 320, let's say, to 200 and something of this phase. You control the diameter. So you might think, who cares? You've gone from 300 to 200. That's not a very big difference. But actually, at this length scale, it makes a huge difference to how they behave. So here's an assay that we run, very typical of the lab. So we have these compartments with fluorescent cargoes inside. It can be DNA. It can be, in this case, it's actually just a fluorescent dye. And you can see, and we basically measure how fusogenic they are, how the ability of these particles to fuse with other membranes. So here we have a synthetic cell membrane, this black membrane, it's a micron scale compartment. And you see in this cartoon here, the small cubosomes, the 130 nanometer ones, fuse perfectly. The larger ones do not. So it's very important to control the size. And this is what a microscopy looks like. You see, when you have the small cubosomes, the vesicles are fluorescent because you successfully delivered the cargo, and the larger ones are not. Then let us ask, okay, this is a cool technique to make lipid nanoparticles of different types. Can we form, the classical thing of a membrane within a membrane? Can you form a compartment within a compartment? You can envisage this being used in drug delivery, right? So say you want to release drug A at time point A, and then release drug B at time point B with two different stimuli. You can have multi-stage dosing. Okay, you can release different doses at different time points. In the case of vaccines, you have to go to the doctor to get vaccine shot 1, and 2 weeks later vaccine shot 2, and not everyone turns up, and compliance becomes a problem, and the whole sort of regime sort of breaks down. Can you design a drug delivery vehicle that can release different doses precisely at different time points controllably, or release, for example, an adjuvant to stimulate the immune response and then the active ingredient? So compartmentalization in principle allows us to do that. So we had to develop a new way to form compartmentalized nanostructures, not microstructures, nanostructures. And the way we did this is again using microfluidics. So we form our internal membrane the same way we always do. And on the surface of these membranes, we have reactive groups. So we have an alkyne group. And then we feed it back through the device. And in the second time we go through the device, you expose it to, if it's an alkyne group to an acyl group, you basically have a click chemistry reaction. They click together, and you basically have very well-controlled assembly of a membrane on a membrane. Control the lipid composition of each membrane to make them responsive to different stimuli, for example. And you can load things in the inner compartment or the outer compartment. So this allows us to form nanoscale compartments. And this is what it looks like. Now, on the cryo, you can only image them on cryo microscopy. They're far too small for traditional microscopy, light microscopy. The cool thing is, you have there's a distribution. So here we're measuring the intermembrane distance. Now, the cool thing is, by playing around with these PEG spaces, you can start to play around with the intermembrane space. And this is what it looks like here. So without any click lipids, you get a bit of a mess. Many membranes assembling one on top of one another. But as soon as you put these click lipids, you start to get these double membrane structures, and you can control the intermembrane space from low to medium to high, shown in this distribution here, simply by playing around with the PEG distance. And again, stimuli responsive systems, we do all sorts of things. So for example, we've designed systems where you can release cargo 1 at stimulus 1, which is temperature 1, and then hit it with stimulus 2, which is a second temperature, and release cargo 2. And that's what's shown in this diagram here. Again, the basis of multi-stage release. Similarly, you can release different cargo. So you can release the blue cargo at time point stimulus 1, and then release the yellow cargo at time point 2. So this is what allows us to release different molecules at different time points. Not only that, you can think of a system which is dormant, again, by separating out the components. It's dormant. You hit it with a stimuli, the compartments break down, they become active, because let's say enzymes start to meet their substrates. You produce an active ingredient, an active molecule, and then you release it. And that's sort of what we've done here. You have an enzyme, you have a substrate, you produce a molecule, you release it. And that's sort of what's shown here. And this was done by Colin Pilkinson, who was a PhD student, postdoc in the lab, and in collaboration with John Seddon at Imperial College. Okay, so I've sort of run out of time. So I'd just like to leave you with 3 take-home messages. Right, the first is, I haven't spoken a lot about the intricacies of the devices. We have some fairly intricate devices now, which allow us to do everything that I presented. But both microfluidics and biomembrane technologies are really powerful tools in synthetic cell design. So many of you work with gene circuits and genetic constructs. Those are very useful, but membranes themselves can also be very useful systems which we could engineer to give us synthetic cells functionality. Membranes and compartmentalization, along with genetics, can be used to engineer synthetic cell behavior, movement, stimuli response behaviors, and so on. And then using similar concepts, using tools and technologies and ideas and concepts that we've developed over the years for synthetic biology, applying them in a different but sort of related area of nanoparticle engineering and drug delivery, really allow us to do sort of different things that weren't previously possible on the nanoscale. So I guess some people say, like, what is synthetic cell? What are they used for? So synthetic cells themselves can be useful, but the technologies used to make them can have all sorts of applications in other areas. And I guess, the stuff I presented right at the end is in a way an example of that. Okay, so that sort of brings me to the end of my talk. I'd like to thank the amazing team. I don't have time to name them one by one, but I'd like to thank sort of the team for some of the amazing stuff that they've done. It's been a pleasure working with everyone since I started my group over the pandemic. And I'd also like to thank the collaborators, current and past collaborators, who are sort of also been sort of invaluable throughout. So that's the end of my talk. I'm going to stop sharing.
Thank you, Yuval. That's awesome. I'm sure at some point we're going to ask you to share again. There are a handful of questions in the chat already. And for the sake of people who are potentially watching this later and can't see the chat, I'm going to ask you to read them out loud before you answer them. But I think we already have maybe 4 questions, and I'm sure there's more coming. Are you willing to jump in?
Yeah. Okay, so the first one from Hendrik. How big are your gap junctions? Okay, so Uh, okay, I don't actually remember from the top of my head, but so how big are they? So what are the dimensions you want? So one of the dimensions is the dimensions between the individual membranes. What is it? What are those dimensions? It's somewhere like, I'm pretty sure, like 4 nanometers or so. So actually similar in length, similar in dimensions to biological gap junctions. The second thing is what size proteins can go through. So what's the size exclusion, I guess, of the membrane? So the things that we use to control what goes through is alpha hemolysin again, a combination of alpha hemolysin and some very clever engine, some very intricate systems where certain pores are blocked and so on and so forth. That's basically alpha hemolysin. So the size is the size of an alpha hemolysin, which is like 1.5 or so, if I'm not mistaken, nanometers. That's what those sort of molecules can pass through, and larger ones can't. Again, it's actually quite similar to the connexin junctions which you see between um, biological membranes. So not proteins, but yes, small molecules. And we're now working so we've shown that we can link up 10, 15 cells together to form a small process tissue. I didn't present it, but we can. But what we want to do is to make masses of them, make like thousands or millions of them to form a proper tissue. So that's sort of the next stage for that sort of piece of work. Okay, so the next question is, did you consider using anything other than lipids, polymers as encapsulation? So yes, we did. We stuck with lipids because we're basically a membrane group. That's all our expertise. What we do is increasingly, I didn't touch on at all, is not polymers. We don't use much polymers, but we use hydrogels a lot, like more and more. So the capsule itself is a hydrogel. And in the hydrogel, you can encapsulate liposomes and vesicles and cell-free and all sorts. And that is becoming really a huge part of the group. And that's led by a fantastic postdoc called Matt Allen. So hydrogels and lipids, those are the 2 things that we use. And hydrogels give you the advantage of being mechanically stable. You can lyophilize them. You can tune the porosity, and are much simpler to work with. So if you want to form a hydrogel bead, you can do that very, very quickly on a microfluidic. If you want to form a giant liposome on microfluidics, it's much more tricky. And also hydrogels, there's a whole decades and decades of research on hydrogel properties that you can leverage, stimulant responsive hydrogels and so on. I think it's very exciting, hydrogel synthetic cells. Okay, the third question again, oh no, by a different Hendrik. Would it be possible to take this work to the next level and combine different types of synthetic cells like synthetic tissues? Yes. So as I said, we do want to form synthetic tissues, but I think what's really exciting here is not different types of synthetic cells, which people are working on, including us, but synthetic cells and living cells to form like a biohybrid. Imagine a synthetic tissue where some of the cells and the tissue are synthetic cells, some are bacterial cells, some are eukaryotic cells, and you have some sort of organoid type system, which is a hybrid. So you can really make the most of the power of biology to photosynthesize, to metabolize things, to grow, to self-heal, self-repair with the programmability of synthetic cells. So we call it sort of cellular bionics or hybrid cells that are part living, part synthetic. That's the interface which we're very excited about, because I started off my talk saying living cells are shit, but actually it's not the case. Living cells are incredibly powerful, and there are certain things which you will never ever be able to do with a synthetic cell, but with living cells you can. So it's the best. It's a matter of taking the best of both worlds, and hybrid synthetic cells is a way to do that. Okay, Mohammed asked a question. What is holding you back most in your research? What is holding back the field most in your opinion, both technically and in other aspects? Okay, so from my perspective, if you look at most of the really cool functions of biology, be it the rotation of a flagellum, be it the way that cells can transduce mechanical information, cannon transduction, or whatever, they're mostly done by membrane proteins. And as you'll see in the presentation, all our proteins are like fake proteins, like alpha-hemolysine. It's not proper membrane protein. We and others have worked with mechanosensitive proteins and so on, but it's basically how to really integrate membrane proteins routinely into synthetic cells. So, what people do is they reconstitute membrane proteins in synthetic cells, but doing it autonomously, where synthetic cell produces its own membrane machinery, is something that I think once we crack that, that will open up loads of new avenues. So from a technical perspective, that's what is holding us back. In terms of other aspects, I don't know. It's an interesting one. One of the people always say, "Oh yeah, like if only people could work better together and be more collaborative." But I actually think the synthetic cell field, compared to other fields, is incredibly collaborative and incredibly open, and it's all just about unlocking that potential. So I don't think there is much holding us back in that sort of front. I think we're doing very, very well on the community front. Yeah. Okay. So the final question, or not the final question, another question. Do you see, when you form membranes in microfluidics, do you see residual carrier phase in your membranes? Okay, excellent question. The short answer is yes. Sometimes we see a pocket of oil which is still there. Mostly they get ejected, but sometimes it's still there. Now, what we wanted to figure out is even if it's ejected and you can't visibly see it, does it affect the membrane properties? Because membranes are so key, does it affect the tension of the membrane, or the permeability of the membrane, or the bending rigidity of the membrane, or all these membrane physical properties which people are interested in? And so we've done many studies. So actually, there's a study which it's not published yet, but we hope to publish soon, led by Ion Ionu, who's a great guy. I don't know if he's on the call, where he's basically investigated this using a host of techniques, including molecular rotors, which basically measure the viscosity of the membrane. And we see a slight difference, but nothing too significant. So the question is, there probably is some residual oil, but we're not worried about it. Whenever we've tried to measure membrane properties using various techniques, we don't see a huge difference. And I think others have put membrane proteins in and they behave fine. So there probably is some oil, but we don't care too much about it now. It's not something that we're worried about or we're trying to fix. Oh God, Charles, can you create 3D nanoscale features by joining lipid subunits or embedding them within a material? Yeah, you should speak to my PhD student who's obsessed with this question. He's just started, so come back in 3 years when he finishes his PhD. Maybe he'll have an answer to you. We haven't tried. The nanoscale stuff is quite new. We embed them in hydrogels, as I said. But whether you could do something more interesting where you could link up individual subunits together to form a material where the hydrogel itself is the bulk material is something that we're interested in, but we haven't done it, the honest truth. But yeah, and as I said, if you look up someone called Bradley on my website and speak to him, he might have some ideas for you. Okay, we have another question from Prathamesh. Their question is about Ola liposomes. How easy were they to make? Okay, this is a $1 million question. So basically, Ola as a technique, can you, as a non-microfluidics lab, use it robustly? So basically, we try. So when we first started working with Ola, we tried and tried and tried and could never get it to work properly. We could form the double emulsions. If you don't care about having an oil in the middle, it's fine. But we could never get the de-wetting. And it was over COVID. So it was an awful time to try and troubleshoot something like this. In the end, we got it to work, and it now works really robustly in our hands because we had some fantastic now postdoc coming from a lab who they did it routinely. So as soon as you have someone who knows what they're doing, it's easy and robust. And we're trying to develop a really robust rule book for others to use. So we very routinely have academic visitors from other labs coming to learn the technique, and they go back to the labs and they're sort of successful in doing it. So it's difficult. So basically, like I don't want to say it's like easy and like you'll be able to do it no problem, just by following a set of instructions. But if you send someone to our lab or a lab who does Ola, spend a few days there and then take it back to your lab, then it's doable. But you need to be really meticulous. Every small change in the humidity and how you dissolve the lipids and how you surface treat it makes a difference. So Ola is something that we're trying to fix, make it more accessible, because at the moment it is quite tricky. I'm not going to lie. But with not everything I presented was Ola. So if you want to make hydrogel capsules and hydrogel compartments, much, much easier. Or if you want to make the nanoscale stuff, also very, very simple. And you'll be able to do it just by reading the protocol of the paper. The older stuff, it's a little bit of an art. So you have to come, learn, and we're happy to host anyone. Okay, Chongyu, are your cells biocompatible, biodegradable synthetic cells in drug delivery? Are they biocompatible basically? So, the lipids are the same lipids that people use for drug delivery. So the lipids themselves will be biocompatible, biodegradable, and the cell-free expression systems also, they're definitely biodegradable, and they probably are, but you might have some sort of immune response, but I don't think that's a big concern for the community, to be honest. So we haven't tried. We haven't done any in vivo work yet. But people have. Avi Schroeder's work in Israel, done a lot of this sort of stuff, and they find that it's not, we're not using some weird rogue chemical or lipid or highly reactive species. So you might get an immune response and that sort of thing, but nothing more than that, I don't think. Yes, our field's collaborative. Thanks to Kay. Yeah, correct. Good encapsulation efficiency. How efficient are the nanocapsules? Okay. Survivability of the capsule is an issue. So yeah, giant vesicles are quite fragile. So, for those of you working with giant vesicles, you form them, you image them on the microscope, they're fine, but you may notice that if you pipette them from one to another, they're still stable. You still get a big population there, but on a microscope, at least, it appears some of them have burst. I don't think anyone's done a systematic study, but membranes themselves can be quite fragile and can burst and reseal and susceptible to osmotic shocks and so on. Now, funnily enough, on the nanoscale, they're stable. They're stable. So you can form them, you can put them in a freezer, you could defrost them, you can transport them, and they're still there at the end. So they are stable. They're chemically linked, they're stable, and it's something to do with the membranes being on the nanoscale makes them more stable. There's no oil destabilizing or anything. So yeah, stability is an issue for synthetic cells in general, but our nanocells are stable. Our micron cells are also fairly stable, but I guess from mechanical and osmotic perturbations, like there's a limit. You have to be fairly delicate in how you handle them. Again, hydrogels give us a solution to that. If you put hydrogels in our synthetic cells, that stabilizes them massively. I think I've reached the end of the questions on the chat. If anyone else has questions, feel free to ask anything else. If not, I just
Yes,
want to
of course.
yeah.
Open to anyone to ask questions. I think I'll give it a last call, but in the meantime, if anyone is thinking of questions, I'm just going to once again plug that we have our Build a Cell workshop coming up on the 20th, held in Boston. These are not available virtually because they are workshops. You have to be there and you have to be working. But we do them twice a year. So if you are interested in that, join our mailing list. And I think maybe we are at the end of our questions. So awesome. I'm going to
Just like
give
to to everyone, if you want to chat or collaborate or send students or whatever, just send an email. We're very friendly, very collaborative. We're in London, easy to get to from anywhere in the world. Great university, very fun bunch of people. So yeah, we're open to anyone really.
I love that. All
Okay.
right. Thank you so much, Yuval, for giving this chat. It was amazing. You covered a lot of ground. You answered a lot of questions for me, and then you sparked a lot more. So I'm excited to go start some reading. And thank
Okay.
everyone else again once more for attending. All right.
Great.
Happy
All right. See
Monday.
you.